In this lecture, we're going to consider the digestive system. This involves the gastrointestinal tract and a range of accessory organs that help the gastrointestinal tract obtain nutrients that we require for our survival. So before we consider the specific anatomy and physiology of the digestive system, let's consider why we need such a system in the first place. Well, our bodies require a constant source of raw materials to survive. We need materials to facilitate the growth of cells, the repair of cells, and also we require the energy necessary to fuel metabolic processes carrying on within cells. However, the raw materials that we require are typically locked up in large food molecules in our diet, and we can't immediately access these essential nutrients. So in simple terms, the digestive system allows us to access those raw materials we need to survive. The digestive system carries out a number of functions. It allows us to ingest food, that means take it into the mouth, transport the food, that is move the food through the digestive system where specific processes can occur. Processes such as digestion. Now, if we talk about digestion, we are referring to the breakdown of large food molecules into small food molecules. Small food molecules can then be absorbed into the bloodstream. What we don't want, we will then expel from the body. If we look at the digestive system, there are two main areas we focus our attention on. Firstly, a large muscular tube called the gastrointestinal tract, also referred to as the digestive tract or the alimentary canal. There are also a range of accessory organs that are attached to this gastrointestinal tract, and they have important roles that we'll speak of soon. This highlights the key structures we'll be considering in this lecture. It breaks up the digestive system into its two main regions. That is, the gastrointestinal tract, so all the structures listed here are associated with the gastrointestinal tract, the long muscular tube that extends from the mouth to the anus. Over this side, we have accessory organs. These are in some way associated with the gastrointestinal tract, and they carry out a function that will help in the processing of the food so we can obtain the essential nutrients. So, key concept, gastrointestinal tract, long muscular tube where the food is, and the accessory organs which are associated with the gastrointestinal tract. This slide summarises what I was just speaking about. So we have the gastrointestinal or digestive tract. There's a continuous muscular tube that extends about 10 metres, and that's in a cadaver. This extends from the mouth to the anus. The key thing is, this is where the food is contained. The gastrointestinal tract includes a range of structures that we're going to talk about, such as those listed here. Pay particular attention to the stomach and the small and large intestine. That's possibly where we're going to spend most of our time. A key concept about the gastrointestinal tract is that there is muscle in the wall of this structure. This facilitates the churning and mixing of food, mechanical digestion, and also the movement of food through the tube, the propulsion of food. We're going to talk about movements such as peristalsis. Accessory organs, as I showed in the previous slide, are associated in some way with the digestive tract, and these include structures such as salivary glands, liver, and the pancreas. They will in some way assist the function of the gastrointestinal tract. So that reminds us of the basic structures we are dealing with. Before we go further, it's important to discuss a basic concept about how the gastrointestinal tract operates. In this structure, digestion occurs, and digestion is the physical or chemical breakdown of food into smaller units. So large food particles being broken down into smaller food particles. Critical to this process is the production of enzymes. Enzymes are protein produced by various digestive system organs, in particular the pancreas, and these act to catalyse or speed up the breakdown of food molecules at the molecular level. So you have large food molecules, complex proteins, complex starch molecules, which are too large to be absorbed into the bloodstream. Enzymes allow you to easily break these molecules down, these large molecules, into smaller molecules, which can be then absorbed and distributed throughout the body. This highlights the basic concepts of digestion. If we talk about mechanical digestion, we talk about physically breaking large food particles down to small ones, such as when we chew food. So we physically break it down. If we're talking about chemical breakdown, we're talking about breaking down food molecules. So we're operating at the molecular level here. So a complex protein is broken down to simple amino acids. This highlights the process of chemical digestion. What we have here is a large enzyme molecule, and over here we have smaller food molecules. Now what you will notice occurring as the cycle begins is a large food molecule, turned to substrate here, will associate with the enzyme. This could be something like a large protein, associates with the enzyme, and as a consequence of this interaction, the larger nutrient molecule is broken down into smaller components. And the idea is that these smaller components should be more easily digestible in that they can be absorbed across. Take 2. This animation here is highlighting the basic concept of chemical digestion. What is occurring is that we have an enzyme here and we have a food molecule associating with that enzyme. Notice how the food molecule will come into association with the enzyme molecule, and then there will be an interaction between these two, and the food molecule is broken down to smaller components, and these smaller components should be easily absorbable across. Take 3. This enzyme is showing the basic concept of chemical digestion. What we have here is an enzyme protein. This is, for example, the types of materials that are produced by the pancreas. There is then a large food molecule that will associate with this enzyme, and during the association, the large food molecule is broken down to smaller components, and these smaller components should be more easy to absorb through the walls of the digestive system and then be distributed throughout the blood to the cells of the body. So digestion is a very important process occurring in the gastrointestinal tract, and it's a key role of the digestive system. Now what we'll do is start considering the anatomy of the digestive system. This shows its overall structure. As I've mentioned previously, it can be broken down into 2 main parts. The first of these is the gastrointestinal tract. That's the muscular tube that involves structures such as the esophagus, stomach, and the intestines. The second major element of the digestive system are the accessory organs such as the liver and the pancreas, and these contribute in some way to the processing of food. For example, the liver and the pancreas provide secretions, they put them into the gastrointestinal tract, and that assists with chemical digestion. This slide just gives us a basic summary about the gastrointestinal tract. Continuous muscular tube, it will extend up to about 10 metres, extending from the mouth to the anus. Has other names such as the digestive tract or the alimentary canal. And the 9 to 10 metres quoted here is what you would find in a cadaver. This is the tubing. So a key concept to get from this slide is that this is all continuous. So if I'm a food particle in here, I will make my way through all of this tubing, unless I'm going to be absorbed through the wall, that is. So let's imagine I'm an indigestible food particle. I will begin here, work my way through the stomach, through the intestines, through the large intestines, and then finally out. The gastrointestinal tract is the site of food ingestion, digestion, absorption, and elimination. And what we'll try and do is highlight what processes are occurring at these individual components of the gastrointestinal tract. Associated with that muscular tube, the gastrointestinal tract, are a number of important accessory structures. These will help the function of the gastrointestinal tract in some ways, typically by assisting the mechanical or chemical breakdown of food. Structures like the teeth and the tongue help physically break down food. Chemical secretions produced by the pancreas and liver and other accessory organs will help break down food molecules. What I'll do now is talk about the basic structure of the gastrointestinal tract. It's a long muscular tube extending from the mouth to the anus. All its length, it has the same basic consistent structure, composed of 4 main tissue layers. The innermost layer we call the mucosa, outside of that the submucosa. Muscularis externa is beyond that, and then the outermost layer of the gastrointestinal tract is a layer we call the serosa. That is if we are below the thoracic diaphragm. Above the thoracic diaphragm, the serosa is referred to as the adventitia. Now if you're in the esophagus, the stomach or the intestines, all basic layers shown here, these 4 basic layers, are present. So it has a remarkably consistent structure throughout its length. Now although these 4 basic tissue layers exist throughout the majority of the gastrointestinal tract, the appearance of each of the layers can vary quite a bit depending on where you are. For example, the mucosa of the esophagus looks quite different to the mucosa of the small intestine. So both parts of the gastrointestinal tract have a mucosa, it's just that in each area the mucosa is quite different. And that difference reflects different functional roles. I'll show you what I mean in the next slide. This slide shows you a magnified view of the innermost layer of the gastrointestinal tract, the mucosa, as it appears in the esophagus and in the small intestine. Now wherever you are, this mucosa will contain an epithelial layer. This epithelial layer is in contact with the food. Beneath that is some loose connective tissue and a little bit of smooth muscle that sometimes helps with the local folding. So a key fact to remember is wherever you are in the gastrointestinal tract, your mucosa will contain these basic elements. Nevertheless, if we look closely at these photographs, we can see some substantial differences in how the mucosa of the esophagus differs to that of the small intestine. Look here at the esophagus. The innermost lining is an epithelium. It's very, very thick, and that suggests that it's protecting the underlying tissue. Here the food particles can be quite rough and unprocessed, so you need this protection from sharper food particles or more abrasive food particles. By the time we get to the small intestine, the food has been liquefied, so abrasion isn't really a problem. What you want to be doing here is absorbing nutrients. There's a lot of chemical digestion that should have occurred by this point, so you want to get hold of those nutrients. So what happens here is that the mucosa has been highly modified. It's folded, lots and lots of folds. These folds are called villi. They provide a huge surface area across which nutrients can be absorbed. So the key role of these structures, these villi, is to increase the capacity to absorb food. So here, the mucosa modified for absorption. Here, the mucosa modified for protection. And this occurs throughout the gastrointestinal tract. So, just summarising that previous slide, it's important to remember that although wherever you are in the gastrointestinal tract there's a mucosa, it's highly modified to suit the function of that particular region. Now, beneath the mucosa, we have the submucosa. That's an easy concept to understand. Sub usually meaning under something. This consists of a loose connective tissue. There we go, that's the layer there, submucosa supporting the mucosa. It's a binding layer. It will link the mucosa to the next layer, which is the muscularis layer, muscularis externa. If you look inside the submucosa, you'll see many blood vessels, highly vascularized. There's also nerves and lymphatics in there as well. The nerve supply, we can call that the submucosal plexus, so these networks of nerves helping to regulate the digestive processes. The next layer out is a muscular layer. Well, it's actually several layers. It's at least 2 wherever you are, sometimes 3 layers of smooth muscle. And the smooth muscle is organised different ways. There is an inner circular layer, so the muscle fibres are sort of surrounding this tube here. And there are longitudinal layers, so the muscle cells also can be aligned going this way. When you have these different arrangements, muscles like this and muscle fibres like that, you can get all sorts of subtle movements, mixing movements and propulsion movements that are very important for the functions of the digestive system. layers of smooth muscle. Within the muscle, you'll also get nerves. We call that the mesenteric plexus. There'll be blood vessels as well in there too. Now, I mentioned before that smooth muscle is found primarily in this layer, the muscularis externa. However, there are the occasional spots where some skeletal muscle can be found. That's something that can be controlled by voluntary action. That involves usually areas of the superior aspect of the digestive tract, near where the mouth is, and at the other end towards the anus. But normally the muscularis externa contains smooth muscle. The next layer of the gastrointestinal tract we'll consider is the serosa or adventitia, the most superficial or outer layer. This is very important when we're looking at the digestive organs in the body cavities. This layer is webbed up to a serous membrane network, helps suspend the digestive organs in the body cavity. Now, they need to be suspended and anchored in place. However, they also have to have the capacity to move because mixing and propulsion of food is an important function of this structure. So we suspend many areas of the gastrointestinal tract through a membrane network. The serosa helps us to attach the gastrointestinal tract to that membrane network. The membrane network I'm referring to is the peritoneum below the diaphragm, the thoracic diaphragm I should say, and we'll consider that very soon. If we're above the thoracic diaphragm, the gastrointestinal tract is lined not by serosa, but a fibrous connective tissue layer we call the adventitia. Now I'll start to consider some of the specific structures of the gastrointestinal tract, and then we'll move on to the accessory organs. Whenever you consider food, the first place you put it is the mouth. So that's where we'll start our examination of the digestive system. The mouth is the entrance to the digestive tract, and it's the food ingestion, it's the taking of food into the body, mastication, that's chewing, and there is a little bit of chemical digestion which occurs as a consequence of materials put in by the saliva. The two regions of the mouth mentioned here, vestibule, that's between the lips and the cheeks, and the teeth and the gingiva, the gums. So this is the most anterior portion. So we won't consider this to any great extent for the rest of this lecture. We'll consider more this region, the oral cavity proper, forming the remainder of the mouth. It's located in front of the oropharynx. I've mentioned the oropharynx before when we did the respiratory system, and the border between the oropharynx and the oral cavity is a fold of the mucosa we call the palatoglossal arch, and we'll see that soon. This highlights some of the structures of the mouth. We'll focus most of our attention on the oral cavity. Oral cavity extending posterior to the gingiva or gums. Note also the teeth. Other structures to note, we can see the tongue that's inside the oral cavity. We can see this conical structure here, the uvula. That's important for closing off entry to the nasopharynx during swallowing. Hard and soft palate forms the roof of the mouth. The hard palate formed by the maxilla and palatine bones. Other structures to note, the fold of mucosa, palatoglossal arch, marks the border between the oral cavity and the oropharynx, which sits posterior to here. Here we also have one of the lymphatic organs, palatine tonsil, that I mentioned when we did the lymphatic system. Structures here, we can see these orifices, openings for saliva, which we'll discuss soon. So saliva enters the oral cavity and helps with the chewing process and basically getting the food ready for it to be swallowed. So a set of accessory organs associated with the mouth are the salivary glands. Salivary glands secrete a fluid we call saliva into ducts that empty into the oral cavity. And we're going to focus on 6 of these extrinsic glands. There are 3 pairs. When we use the term extrinsic, we mean outside of the area of interest. So these salivary glands are located outside of the oral cavity. They are linked to the oral cavity by tubes or ducts, and the saliva they produce enters the oral cavity via that ducting system. Now, saliva contains a number of components that help you process food. There's mucus and fluid in it that helps lubricate food, helps you chew it up and get it into a paste-like consistency so it's easier to swallow. Amylase is an enzyme that breaks down carbohydrates, so carbohydrate digestion can commence at this point. There is also the capacity of the saliva to disinfect the food, because when you put food in your mouth, you're giving the opportunity for pathogens to enter your body. So by putting in antibodies and enzymes to the saliva, you can inhibit the growth of bacteria and other microorganisms. Now there are 3 pairs of extrinsic salivary glands, and they are indicated here. 1, 2, and 3. 3 on each side of the face. The largest of the salivary glands are the parotid salivary glands, indicated here, located anterior and inferior to the ear. And these produce about 1/4 of the saliva. Inferior to the body of the mandible, or the jawbone as we commonly refer to, this is the submandibular salivary gland, and this produces the majority of the saliva. Inferior to the tongue, as indicated here, we have the relatively small sublingual salivary gland, and these contribute only about 3 to 5% of the total saliva. So just remember that there's 3 of these on each side of the face. 3 pairs of extrinsic salivary glands. Now, since these salivary glands are extrinsic, which means they're outside the oral cavity, they have to get their saliva in via a system of ducts. So here we have the parotid salivary gland. Linked to that, we've got the parotid duct, and that will take the saliva into the mouth, posterior to the molar teeth on the upper part of the mouth. The submandibular gland here, that has a gland that takes the saliva into the oral cavity just behind the incisal teeth. The sublingual salivary gland puts its saliva into the oral cavity via a series of small ducts that we can see here. So that is how the saliva gains entry to the oral cavity. And let's not forget that there are 3 glands on the other side as well. I should mention here that the parotid salivary gland, this is the one that becomes swollen during mumps. So if you ever had mumps, this is the part that got swollen. So what should be happening in your digestive system? You put the food in your mouth, you chew it up, the tongue, the teeth help physically break up the food. The saliva gets poured in, and when that saliva pours in, the food can become lubricated, a bit of amylase there to help break down the carbohydrates, a bit of disinfection occurs. Then you're ready to move the food to the next part of the digestive system. That's the pharynx. Now the pharynx we've touched on before when we did the respiratory system. The pharynx is associated with both the respiratory and digestive systems, commonly referred to as the throat. Now the entire pharynx originates posterior to the nasal cavity and extends to the point where the larynx and esophagus go in their separate directions. The pharynx consists of 3 key regions which we've looked at previously, so I won't spend too long on this. There's the nasopharynx, posterior to the nasal cavity. That has no involvement in the digestive system at all. Oropharynx sits posterior to the oral cavity, and inferior to the oropharynx we have the laryngopharynx. The laryngopharynx is continuous with the esophagus. And here we're just briefly looking again at these specific regions of the pharynx. We've touched on these in a bit more detail when we looked at the respiratory system, but we'll emphasize it for you again. Here we have the nasopharynx, posterior to the nasal cavity. There's the opening to the auditory tube that links to the middle ear. There's the adenoids there, tonsils. Posterior to that we have the oropharynx. Now the oropharynx allows movement of air and also food and liquid, so it's a component of both the digestive and the respiratory systems. We then have the laryngopharynx inferior to that. Laryngopharynx continuous with the esophagus. Notice here that we're splitting the systems up. We have the tubing going down here. There's the larynx there, part of the respiratory system. At this point, that is just dedicated to the movement of air. there should be no food entering this larynx structure. On the other side here, we have the esophagus. This is dedicated towards the movement of food towards the stomach. So in these two regions of the pharynx, we can service the movement of food, liquid and air. When we get to this point, however, air goes into the larynx, food goes into the esophagus. And this just highlights some of the concepts I was speaking of in the last slide, so I won't go too far into the detail listed here because we've already covered that. Nasopharynx, most superior region of the pharynx. Oropharynx, posterior to the oral cavity, extends inferiorly to the body of the hyoid bone, which is at about the level of cervical vertebra number 3. We then have the laryngopharynx, which is inferior to the oropharynx, and that extends to the point where the larynx and esophagus goes in separate directions. So the laryngopharynx, directly continuous with the esophagus. Anterior to the esophagus, you have your larynx. The larynx carries the air, esophagus carries the food and the liquid. Laryngopharynx links with the esophagus at about the level of cervical vertebra number 6. In this slide, we have shown for you how we prevent food and liquid entering the airways. So the green material here is a pasty lump of food we call a bolus. We've chewed it up and we're ready to swallow it. So now we're swallowing the food. Look what's happened to prevent food going the wrong way. Here we have the soft palate and the uvula closing the entrance to the nasal cavity, so food should not exit this point. Over here, the epiglottis has come over the top of the larynx and food cannot enter this tubing. If it does, then you've got major problems. Food is instead directed down the esophagus towards the stomach. So food and liquid should follow this path, and the inappropriate movement of food into the nasal cavity or the larynx is prevented by these specific valve systems. As we project inferiorly from the pharynx, we will eventually hit the esophagus, a muscular tube, and this will transfer the food and liquid from the laryngopharynx to the stomach. 3 key regions of the esophagus associated with the part of the body it's located. The most superior component is the cervical esophagus, continue with the laryngopharynx in the cervical region of the body. Then you have a portion that runs through the thoracic cavity, the thoracic esophagus, and then we have a short portion, the abdominal esophagus, which projects below the thoracic diaphragm to attach to the stomach, and that's typically only about 1 centimetre in length. The movement of food down the esophagus towards the stomach is achieved by a wave-like motion we call peristalsis. And this peristalsis is occurring because there's coordinated contraction of muscle fibres in the muscularis externa of the wall of the esophagus. So by carefully coordinating the contraction of the different muscle layers in the esophagus, food is propelled down in a wave-like motion. And this is what I'm talking about. So here's food movement in the esophagus. There's the esophagus there. The muscularis externa layer has layers of muscle, and when this contracts, if you coordinate it the right way, you get this wave-like motion occurring, propelling the food down towards the stomach. So the food just doesn't flop down by gravity, it's propelled by muscular contractions. As we approach the stomach, there's a muscular valve we call the gastroesophageal sphincter or cardiac sphincter. It will relax and allow the food to enter. What I'm going to do now is show you an animation which briefly illustrates the process of peristalsis occurring in the esophagus. So concentrate on the wave-like motion. So there we could see that the movement of food down the esophagus is an active process requiring the careful coordination of muscle contraction to achieve peristalsis. So there's a lot of movement going on in the digestive system, propelling of food forward, such as we saw there, and also the churning and mixing of food, as we might see in the stomach or in the intestines. Now once you get down to the level of the stomach, you are in the abdominal pelvic cavity. And in this cavity there is a network of membranes that helps to suspend the digestive organs into position. This membrane network is referred to as the peritoneum. So, quick definition here: abdominal pelvic cavity, extending from the thoracic diaphragm to the pelvic floor. We covered that in the first lecture. Within this cavity is a membrane of serous membranes, so membrane network. These suspend the digestive organs into position. And we've seen serous membranes before when we were looking at the pleural membranes of the lungs. Now, serous membranes are characterized by two key layers: a parietal layer, which lines the body cavity wall, wherever you are, and then another layer, the visceral layer, that lines the actual organ being covered. These two layers join with each other, so it's a continuous membrane network. Between the two layers is a space, and there will be fluid in that space, and that concept was highlighted when we looked at the lungs. That fluid helps reduce friction, which is important when you're dealing with organs such as the lungs or the digestive organs that are capable of movement. This is showing you the peritoneum in the abdominal pelvic cavity. Let's focus on this image here first, the fist in the balloon. This highlights the two-layered nature of the peritoneum. We have a layer of the balloon here coating the hand. That's equivalent to the visceral layer of the peritoneum. It's continuous with this outer layer. That's equivalent to the parietal layer of the peritoneum. So that layer here would be in contact with the body wall. And here we can see those elements of the peritoneum membrane network highlighted. Parietal peritoneum lining the body cavity wall. Visceral peritoneum lining the actual organs. Now what you get between them is often this material here, what we call mesentery. And that's where there's two layers of serous membrane that are formed. Mesentery is very important for linking these various aspects of the peritoneum together, and also for allowing access to blood vessels, nerves and lymphatic vessels so as they can get to these digestive organs. So key thing to note: serous membrane network, parietal layer lining the body cavity, visceral layer lining the individual organs, and mesentery connecting these layers. Notice also that some of the organs in this cavity exist outside of this membrane network, and we refer to those as being retroperitoneal. Organs such as some parts of the large intestine, the duodenum for example, is also retroperitoneal. Kidneys are too. So not all organs within the abdominal pelvic cavity exist within this network, but many of the digestive organs do. Now in the last slide I showed you a sheet of material I referred to as mesentery associated with that peritoneum. Mesenteries are double layers of the peritoneum. They help to suspend the abdominal viscera in their correct location, so the organ is located where it should be. But because it's suspended, it can move, and that's important if you're churning food or propelling food. The mesentery also gives an opportunity for blood vessels and nerves to access the digestive organs. Two of the mesenteries that are worth bringing to your attention are the greater and lesser omentum. The greater omentum is easy to locate. It's a very large fold of the peritoneum. It extends from the inferior aspect of the stomach, suspends itself anteriorly over the small intestines, then it loops back on itself, heads towards the transverse colon, and then it will project to the posterior abdominal wall. So it's like a curtain of membrane hanging over the front of the small intestines. The lesser omentum is also easy to locate. It extends from the inferior aspect of the liver to the superior aspect of the stomach. This is highlighting those two mesenteries that I spoke of. So the lesser omentum links the liver to the stomach. So the inferior aspect of the stomach, lesser curvature of the stomach, linked by this lesser omentum. This other sheet of mesentery is the greater omentum, attaches to the greater curvature of the stomach, draped over the front of the small intestine, reflects back upon itself to about, and then projects upwards to the level of the transverse colon, then projects towards the posterior body wall. Now that we're in the abdominal pelvic cavity, I'll start considering some of the digestive organs that are located there. The first of these is the stomach. This is the one that links to the esophagus. It's a C-shaped enlargement of the gastrointestinal tract. So it's really just a highly dilated region of that gastrointestinal tract. And it will hold varying volumes of material in it. So it might only be 50 mils if the stomach's relatively empty, or if you've had a good feed, you might have to have up to 4 litres of capacity within it. So there'll be 4 litres worth of food being digested in it. What goes on in the stomach is we start to liquefy the food. So the food enters as a paste and exits as a fluid, and the fluid is much easier to chemically digest than a paste would be. So in the stomach, you hold the food, you mix it with gastric secretions, fluids and other materials secreted by the cells of the stomach. The material, the food, then becomes liquid. There's also a bit of digestion occurring at this point, a bit of protein and fat digestion. The liquid that we create in the stomach is called chyme. The processes that occur in the stomach are facilitated by the muscular movements. There's a lot of muscle in the stomach. There's actually 3 layers in the muscularis externa. A lot of churning can occur there. And the other key thing that goes on in the stomach, as well as the churning, is the secretion of the gastric juices. So by the time, after a few hours of holding the food there, you should be ready to move the food forward into the intestines, and at that point it should be very liquid, what we call chyme. This shows the gross anatomy of the stomach, and I'll just highlight a few of the main features now. So we have a greater and lesser curvature. The lesser curvature is associated with the lesser omentum here, and the greater curvature here associated with the greater omentum. So it's a curved structure. We have a superior dome-shaped segment. We call that the fundus. And a fundus was also seen when we looked at the uterus, or will be looked at when we look at the uterus. Notice the thoracic diaphragm here. We're now in the abdominal cavity. Note the 3 layers of muscle in the wall of the stomach: longitudinal, circular and oblique layers of muscle. This allows a lot of mixing and churning and propulsion of food to occur. As well as the fundus, we have the main body of the stomach, and then we have this funnel-shaped portion we refer to as the pylorus. The chamber here, the pyloric antrum, and the canal here that marks the border as you're starting to progress towards the small intestine, that's referred to as the pyloric canal. A muscular valve here, the pyloric sphincter, controls the release of fluid from the stomach into the small intestine. So this is the small intestine here. Notice also the extensive folding within the stomach, rugar. This allows for expansion of the organ as food enters it. So wherever you have rugar, this organ will be able to expand. Here I'm summarising some of the structures that I was just speaking about. The 2 curvatures that mark the superior and inferior border of the stomach, the dome-shaped fundus, the funnel-shaped pylorus adjacent to the small intestine, the main body of the stomach, and the rugar, the folds that facilitate expansion of the stomach. So after being in the stomach for a few hours, the food is ready to be moved into the small intestine. At this point, the food should be quite liquid. The small intestine extends for about 6 meters in a cadaver, but it's shorter in a living individual. The small intestine extends from the pylorus of the stomach to the cecum of the large intestine and takes up a significant portion of the abdominal cavity. There are 3 key regions: duodenum, that's proximal to the stomach, about 25 centimeters, jejunum, about 2 meters, and the ilium, 2 to 3 meters in a living individual. A really important thing that occurs here is the absorption of nutrients. Before that can occur, a lot of chemical digestion will occur, so fluids from the liver and the pancreas that assist in chemical digestion will enter the small intestine, break down the food molecules, and then those food molecules are absorbed in the small intestine. Your nutrients will spend at least 12 hours in the small intestine to be chemically broken down, and then they can be absorbed. This shows the location of the small intestine in the abdominal pelvic cavity. So there it is there. This is the large intestine there. There's the pancreas and there's the stomach. So linked to the stomach we have the duodenum, the first part of the small intestine, and then the rest of it is here. And then that joins on to the cecum about here. On this side what we've done is stretched out the small intestine to highlight its major regions. We've also shown the adjoining structures. There's the stomach, and this is the large intestine. So between those two, the stomach and the large intestine, is the small intestine. Duodenum, proximal to the stomach. Jejunum, that's about 2 meters, and then we finally have the ilium, 2 to 3 meters. That links onto the cecum here. So in this coiled tubing, what will be occurring is a lot of chemical digestion, food molecules being broken down to forms that can be rapidly absorbed through the body wall. So the wall of this intestine, the nutrients will have to be able to absorb through it. They have to be small enough and accessible so they can be absorbed through the wall of the intestine and picked up by the blood supply. So chemical digestion and nutrient absorption are the two key roles of the small intestine. Really important concept, that. This summarizes what I was saying. Small intestine is the major site of food digestion. The food molecules are broken down to sizes which are small enough to be absorbed across the wall of the intestine and enter the underlying blood vessels. Emphasize again the 3 regions of the small intestine: duodenum, jejunum and ilium. Now an important concept to note is that if you look carefully at the wall of the small intestine, you'll notice it is highly folded. The folding occurs at multiple levels. There are quite large folds we call plicae circulares. These are folds of the mucosa and submucosa. They are visible without the aid of magnification, and you'll find a lot of those in the duodenum. Then if you look more closely and have the capacity to magnify, you will notice folds of the mucosa that are called villi. They're about a millimetre or so in height. You get about 20 to 40 of these per square millimetre. The epithelial cells that line the small intestine, if you look at high-powered magnification of their membranes on the luminal surface, you will notice they are highly folded as well into what we call microvilli. The key outcome of all this folding of the villi and the microvilli in particular is that it vastly increases the surface area of the small intestine. So you've got a much greater capacity to absorb nutrients. The more folding, the more absorption can occur because the surface area is bigger. Now this is just highlighting the concept of the folding that exists in the small intestine. So once again, previously I mentioned differences in the appearance of the mucosa in different regions of the gastrointestinal tract. Those differences relate to what's occurring there. The mucosa in the small intestine appears this way, highly folded, because it reflects what's going on there. First of all, we have the plicae circulares, which are visible without the aid of magnification. These are particularly prominent in the duodenum. Then we have folds of just the mucosa, we call villi. These create a very high surface area, and nutrients can be absorbed across that surface area. That huge surface area allows you to capture lots of nutrients. Notice the underlying lymphatics and capillaries, blood vessel capillaries, through which nutrients will be picked up and transported. So if we look closely at the epithelial cells that line the villi, we notice that their cell membranes are highly folded. This also increases the surface area across which nutrients can be absorbed. So a key concept: lots of folding of the mucosa and submucosa in the small intestine, and also folding of the cell membranes of the lining epithelial cells, altogether vastly increasing the surface area available for absorption. Now what we'll do is show you briefly an animation of the villi of the small intestines and the nutrients being absorbed across the surface of them. So this shows the walls of the small intestine, and what we can see here are the villi projecting outwards. These are about 1 millimetre high, and you get between 20 to 40 of these per square millimetre. We'll run the animation now, and we'll pull out one of these individual villus projections. There it is there. Now we'll section it, and what we've got here are the underlying capillaries and lymphatic vessels. What will appear over here are nutrient molecules. These are small enough to pass across the membranes here of where the epithelium is through the underlying connective tissue and be taken up into the circulation system. So blood vessels and lymphatic vessels will take up the nutrients. The nutrients have been chemically digested, so they're small enough to enter this system. In this section, I'll consider some of the specific components of the small intestine. Proximal to the stomach, we have the duodenum. This curves around the head of the pancreas. It's relatively short, only about 20 to 25 centimetres in length, and it's retroperitoneal, which means it sits behind the peritoneum membrane network. Distal to the stomach, this will connect with the jejunum. You can see the folds here, prominent folds of both the mucosa and the submucosa. They are the plicae circulares. This is the point of reception for fluids coming from the stomach. It's also the point where we add secretions that are being produced by the pancreas and the liver into the cavity here where the food is. The jejunum is continuous with the duodenum. This extends for about 2 metres. It's a primary region for chemical digestion and nutrient absorption. So secretions from the pancreas and the liver will help to break down food molecules to forms that are small enough to be absorbed across the lining of the gastrointestinal tract. The most distal region of the small intestine relative to the stomach is the ilium. It can be up to 3 metres long. The distal end of this structure terminates at the ileocecal valve. That's a muscular valve that controls the entry of the material into the large intestine. And the point of the large intestine is to process waste. So just to remind everybody that a key role of the small intestine is to digest the food and absorb the nutrients. Once the food moves out of the small intestines, hopefully most of the nutrient digestion would have already occurred. And the final portion of the gastrointestinal tract that we'll consider is the large intestine. This is approximately 1.5 metres in length, that's in a cadaver, and is characterised by muscular pouches we call haustra. The key role of the large intestine is to prepare the waste material for removal from the body. And one of the key things we've got to do in that process is remove the water. So what will happen is about 500 mils of residue from the small intestine will enter each day. The entry of that material will be via the muscular ileocecal valve. Over time, the material, the water from that will be removed as much as possible. And basically what happens is about 350 ml of fluids and associated electrolytes will be removed from every 500 mils of residue that's come in from the small intestine. So for every 500 mils that enters, only about 150 grams will end up leaving the body. And the material that leaves the body is referred to as feces. The large intestine has several specific components. The cecum is the first of these that we'll consider. This is the one that attaches to the ilium. It's a blind pouch in the lower right abdominal quadrant, and it sits below the ileocecal valve. Inferior to that is the veriform appendix. Now that's a lymphatic structure that's involved with providing you with immunity from disease. It has no role in digestion. The cecum is intraperitoneal, so it is within the membrane network of the peritoneum. Superior to the cecum we have the ascending colon. This extends up to a point where it turns at 90 degrees, and this is the right colic or hepatic flexure. The hepatic referring to the proximity of the liver, which is superior to this. Retroperitoneal, so it sits outside the membrane network. The transverse colon then extends horizontally from the hepatic flexure to the next flexure, that's the next 90-degree turn, and that's either splenic or left colic flexure. Transverse colon is within the peritoneum network, intraperitoneal. So this is showing you the large intestine. Note its major components. Here is the ileum, and there's the ileocecal valve. There's the appendix, I've mentioned all these. Inferior to the ileocecal valve we have the cecum, pouch-like structure there. Superior to that we have the ascending colon, 90-degree turn at the hepatic flexure, horizontally projecting transverse colon to the splenic or left colic flexure. Another 90-degree turn that will project inferiorly down the descending colon, and there's the S-shaped sigmoid colon. Feces is then stored in the rectum and passed out of the body via the anal canal. We'll consider these structures in a few minutes. Other structures to draw your attention to are the individual haustra. So here we've got a haustrum, and there are multiple haustra, these pouch-like structures that help with the waste processing. They help squeeze the fluid from the waste. Tinea coli is this strip of muscle that's indicated. That's just a thickening of the longitudinal muscle. Also notice the mesentery sheets providing access for blood vessels here. And this highlights the regions of the large intestine that I haven't mentioned yet, but I pointed to into the diagram. Descending colon extending inferiorly from the splenic flexure to the sigmoid colon. That's retroperitoneal, so it's not in the peritoneal network. The S-shaped sigmoid colon. The rectum, this is the point where we store the feces just before it's removed from the body. Now these have folds, rectal valves, horizontal folds of the mucosa that help to prevent the feces leaving the body until it's ready for removal through defecation. So if gas is passed, the rectal valves prevent the feces coming out. When feces is removed, it's passed through the anal canal. That's the final 3 centimeters of the large intestine that terminates at the anus. And the passage of the feces through this material is regulated by a couple of layers of muscular valves. There's an internal and external valve, and it's the external one, made of skeletal muscle, that you control through voluntary control. So you consciously control the release of this valve so you can determine the point of defecation. This shows the rectum and the associated anal canal. The rectum is continuous with the sigmoid colon. Feces is stored here prior to defecation. Notice the folding here, the rectal valves, which will retain feces if gas is being passed. When defecation occurs, feces is then passed through the anal canal out via the anus. Muscular valves control the release of the feces from this region. Smooth muscle for the internal anal sphincter. The relaxation of that is controlled by a reflex action. The other one here, the external anal sphincter, relaxation of that is voluntarily, so you decide when this is relaxed. In this last section of the lecture, I'll consider a couple of the most important accessory organs of the digestive system, the liver and the pancreas. These are epithelial glands that produce important secretions. They're important for chemical digestion within the small intestine. The liver plays many roles in the body, but its role in the digestive system is to secrete bile that breaks down fats. And it breaks it down similar in a fashion to detergent, so large fat droplets broken down to smaller fat droplets. These are then more accessible to enzyme, which will break down fats that are produced by the pancreas. Pancreas has a key role in producing enzymes that break down food at the molecular level. The pancreas also produces bicarbonate ions which neutralize stomach acid. So the first structure I'll consider is the liver. It's an epithelial gland. It's the largest gland in the body. It's located in the right upper quadrant of the abdomen, just beneath the thoracic diaphragm. Has many roles in the body. We're only really interested in its role in digestion in this lecture. What it does is produce bile that breaks down fats into small droplets. This will assist in the chemical digestion of fats, because if you have small droplets, they are much more accessible to fat degrading enzymes produced by the pancreas. Other roles of the liver include detoxifying drugs, metabolites and poisons. You can store nutrients in the liver. In particular, glucose is stored in the liver in the form of a molecule we call glycogen. Liver is also involved in producing various blood proteins, and there are cells called Kupffer cells within the liver that help clean out materials from the blood. We'll have a brief overview of the anatomy of the liver. So some of the most important structures are mentioned here. There are a couple of very dominant lobes. There's a left lobe and the right lobe. The right one is the largest of the liver lobes, and the left lobe is separated from the right lobe by a sheet we call the falciform ligament. It's a sheet of mesentery. There's a hilum in the liver. We call it the hepatic hilum, hepatic a reference to the liver. So, as in other areas that we've looked at, like the lungs or lymph nodes or other structures, where we have a hilum, it's a point often where vessels or other structures enter a particular organ. Just beneath the liver, we also have a bladder. It's called the gallbladder, and it's important for storing bile, which is secreted by the liver. So it stores it and concentrates it, and then it releases it into the small intestine. This shows an anterior view of the liver. Take note of the right lobe and the left lobe, separated by the falciform ligament, that sheet of mesentery that I referred to. Notice the gallbladder, which is inferior to the right lobe. We've also got a structure here indicated called the round ligament. That's just like a fibrous remnant of the umbilical vein. This shows an inferior view of the liver, and the main lobes are indicated, the left one and the right one. Notice also this sort of sheet-like structure there. That is the peritoneum. Covers the majority of the liver, but there is a bare area here. Inferior vena cava here, major vein of the body, and here we have the hilum. This is the area where blood vessels are entering or exiting. We have the hepatic artery proper, which is delivering oxygenated blood to the liver. We have the hepatic portal vein, which is delivering nutrients recovered from the small intestine, and we have here ducts which carry bile that's been secreted by the liver. The gallbladder here will store that bile, concentrate it, and then control the release of it into the small intestine. This slide here shows a problem that can occur in the gallbladder. Unfortunately, chemicals in the bile can come out of the solution sometimes and form sharp stones. This can obstruct bile flow from the liver, and also it can cause intense pain when the gallbladder contracts to try and release the bile. See the gallstones here? You would treat these with lasers, drugs, or surgery. The transport of bile is facilitated by the biliary system. It's a series of ducts that take bile towards the small intestine. So bile is secreted by cells in the liver, and the bile initially passes out of the liver via a couple of main ducts, the right and left hepatic ducts. These link, they converge to form a common hepatic duct. This projects down until a point where two other ducts diverge from it. One is the cystic duct. That heads towards the gallbladder, and this will carry bile secreted by the liver, and the bile will be delivered to the gallbladder where it's stored and concentrated. The other ducting that I was talking about is the common bile duct. This is at the union of the cystic duct and the common hepatic duct, and this will take bile towards the small intestine. So the common bile duct will project down actually towards the pancreas, and just before it reaches the small intestine, it fuses with another duct that is coming from the pancreas. So you get this fusion of ducts from the pancreas and the liver. There's a dilated region where this convergence is evident, the hepatopancreatic ampulla. Hepato, reference to the liver, pancreatic, reference to the pancreas, ampulla, dilated region. At this point, the materials produced by either the liver or the pancreas can then be emptied into the duodenum. The structure where this occurs is a nipple-shaped structure, the major duodenal papilla. This is showing you the ducting system associated with the transport of bile from the liver. These two ducts, these are the right and left hepatic ducts. Above here we would have the liver. They fuse together to form a common hepatic duct. At this point here, there's this branch we call the cystic duct, takes bile to the gallbladder. Gallbladder holds it, concentrates it, and then releases it into another duct we call the common bile duct. Common bile duct then projects down through the pancreas, and then just before it reaches the small intestine, it fuses with the main pancreatic duct, as you can see here. This main pancreatic duct and common bile duct fuse together and form a dilated region, and this is referred to as the hepatopancreatic ampulla. This nipple-shaped projection here, major duodenal papilla, marks the point where the bile from the liver or the material from the pancreas is released into the duodenum. The pancreas is what we call an epithelial gland, and it has mixed functions. It has endocrine functions, has very important roles in controlling the level of blood glucose. We're not going to consider this. This involves the production of enzymes such as insulin and glucagon. We're not interested in this function in this lecture. We're interested in this, the exocrine functions. This relates to the production of materials that will be carried along duct systems. The pancreas helps the digestive system because it produces digestive enzymes that break down lipids, carbohydrates and proteins. Important nutrient molecules, these are broken down at the molecular level by enzymes produced by the pancreas. The pancreas also produces substances to neutralize the stomach acid. The secretions produced by the pancreas enter the small intestine at the duodenum, at that structure I showed you before, where there was convergence of the main pancreatic duct and also the common bile duct. The pancreas is a retroperitoneal gland, which means it sits outside of the peritoneum membrane network, characterized by a head, body and tail. The head is adjacent to the duodenum and the tail is in close proximity to the spleen. Ducts carry the digestive enzymes and bicarbonate ions to the duodenum. There are a couple of ducts in the pancreas. I've already shown you the main pancreatic duct. That is the one that fuses with the common bile duct and puts its secretions into the duodenum at the greater duodenal papilla. There is also a smaller accessory duct that enters into the duodenum superior to the main pancreatic one. In this final slide we can see the gross anatomy of the pancreas, the leaf-shaped structure. Here's the duodenum, head of the pancreas here, body and tail of the pancreas there. Notice the main pancreatic duct fusing with the common bile duct, and we saw here the secretions from both organs entering the duodenum. Notice also this other accessory pancreatic duct. Over here we have some histology, some of the cellular structure of the pancreas, and this is simply put here to highlight the fact that this organ has two key roles. It has patches of tissue in it that we call the islets of Langerhans or pancreatic islets. They make hormones that are important for the regulation of blood glucose. The brown material, not the red stuff, but the brown stuff, pancreatic acini, that is the part of the pancreas, the tissue in the pancreas that manufactures the digestive enzymes and the bicarbonate ions. So it's a very, very useful organ, the pancreas, regulating blood glucose and also making major contributions to the digestive system. That's it for this lecture. In the handout that I've given you, there are some additional slides with lots of models, with lots of labels. That's primarily to help you identify structures in the practical that you'll be attending related to this topic. That's it. Thank you.